Welcome, everyone, to today's session of Global IMMUNOTALKs, the series that brings all aspects of immunology to a global audience. Now, today we welcome Mark Cragg. Mark Cragg graduated from Bath University and went on to do a PhD in Southampton, after which he spent some time as a postdoctoral fellow at the Walter and Eliza Hall Institute in Melbourne, Australia. He then returned to the UK in 2007 and went back to Southampton to set up his research group. Now, he's currently a professor of experimental cancer biology in the School of Cancer Sciences at University of Southampton. And in 2024, he was elected a Fellow of the Academy of Medical Sciences, a very notable distinction in the UK. Now, Mark's research is concerned with how cancer drugs can be made to destroy tumors and with a particular focus on antibodies. And his recent work, which he will be telling us about, concerns the engineering and optimization of agonistic antibodies against receptors, in particular of the TNF receptor family for cancer immunotherapy, and also the interplay with FC receptors. Now, as usual, the talk today will be followed by a Q&A session, which will happen offline on X, and the instructions will be put up on your screens at the end of Mark's talk. And so in a minute, I'm going to ask Mark to deliver his lecture. But as usual in this series, I'm going to first ask Mark a personal question, and that is, Mark, in what ways has interdisciplinary collaboration enriched your scientific perspective?
Yeah, thanks, Katano. I think you'll see from the talk, I think bringing together people from different disciplines just enriches the depth in which you can understand a particular scientific problem, and the kind of things that we're trying to do with antibody engineering requires people from structural biology, computational chemistry to come together to really be able to get deep insights into how we engineer these antibodies for better efficacy. So it's been really rewarding to see how different people come together to try and solve a common problem.
Very good. And so on that note, I'm going to ask you, Mark, if you can start sharing your screen and deliver your talk, which is entitled Engineering Antibodies for Immune Stimulation. So, Mark, the floor is yours.
Fantastic. Hopefully you can see that slide.
We can, yes.
Fantastic. So yeah, as Katana mentioned, I'm going to talk about how we go about engineering antibodies for immune stimulation. So this work is done in the Centre for Cancer Immunology in Southampton in the UK. And really it's in the context of cancer immunotherapy, which I guess, prior to maybe a decade or so ago was not seen as something that was particularly likely to have great inroads into cancer. But I think in the last decade, certainly, we've been able to show that cancer immunotherapy is a really valuable and increasingly sought after modality of trying to treat cancer more effectively. So it was labeled as Breakthrough of the Year back in 2013, and I think since then it's gone from strength to strength. So that involves different types of immune cells being leveraged against cancer. But I think perhaps the biggest advances have come in the context of antibody immunotherapy. So these are your classical Y-shaped molecules. We have two Fab arms that do the binding to our particular targets, and then we have a molecule at the bottom part of the molecule called the FC domain. And really the types of molecules that we use, the types of antibodies are really in three main types. Direct targeting are molecules such as rituximab or Herceptin that you may have heard of that bind to the tumor cell itself and cause its destruction in some way. And a kind of direct relation to those are the antibody drug conjugates, where we have the direct targeting part of the antibody taking a toxin of some point, of some type, to the tumor cell to leverage destruction. But what I'm going to talk about, and really where I think the excitement in the field has built really, is around immunomodulatory antibodies. So these antibodies don't bind directly to the tumor, they bind to cells of the immune system typically. And again, they come in a variety of different flavors, particularly two types, either checkpoint blockers or immunostimulatory antibodies. And the ones that have really translated into the clinic so far have been the checkpoint blockers. So these are ipilimumab and nivolumab. So binding to checkpoint molecules such as CTLA-4 and PD-1. And they've certainly revolutionized our ability to treat certain types of cancers more effectively. You can see the survival curve there on the right. But unfortunately, even these reagents have limitations in their efficacy, and there's also toxicities associated with some of their use. So we need to keep finding other ways of augmenting immune suppression, of overcoming immune suppression and resistance that these checkpoint blockers have. And what we consider to be the most likely and tractable method would be to engage immunostimulatory antibodies or ISAs. So these antibodies, rather unlike checkpoint blockers, which are essentially there to prevent interaction of an inhibitory ligand with a receptor, basically these do the opposite. They bind to stimulatory receptors, and they're there really to boost anti-tumor immune responses to overcome immune suppression. They typically target co-stimulatory receptors such as tumor necrosis factor receptors, expressed on immune cells to elicit agonistic effects, so stimulatory effects on those cells. And that can result in T cell activation, proliferation, secretion of pro-inflammatory cytokines, and ultimately anti-tumor responses. And obviously, all of these immunotherapies can give us the capacity to have memory, which can help prevent relapse and recurrence. And the challenge really here is to make powerful agonists that can target these co-stimulatory receptors and then target them appropriately. And the paradigm for what we've worked on predominantly in Southampton has been targeting the TNF receptor CD40. And the reason for that is because CD40 kind of sits at the heart of the immune system in terms of it's part of the humoral immune system in terms of generating appropriate class switching and antibody production, but also it's a key receptor on antigen presenting cells for then licensing those antigen presenting cells to then license cytotoxic T cells to go off and kill tumor cells. And over 2 decades ago, Martin Glennie's group in Southampton showed that targeting CD40 with agonistic antibodies in mouse preclinical models could be effective in at least controlling and deleting some of our preclinical tumor models. So it's always been an exciting potential target. And when I got involved in the project, really, I was involved in understanding how Fc gamma receptors are important for determining antibody immunotherapy. So for those of you who aren't aware, Fc gamma receptors bind to IgG components of antibodies, and then they contact basically with various immune effector cells. They come in 2 flavors, activating Fc gamma receptors and a single inhibitory receptor called Fc gamma receptor 2 or 2b or CD32b, and that's an inhibitory receptor. And one of the things that we were doing at the time was trying to understand which Fc receptors should we engage. And the simplest way of trying to do those studies preclinically were to use 2 different isotypes of antibody in the mouse. So the mouse IgG1 isotype is known to bind strongly to really only to the inhibitory Fc receptor, whereas the mouse 2A isotype binds very strongly to various activating Fc receptors. So it was quite a good way of seeing whether we needed activating or inhibitory Fc receptors for therapeutic efficacy. And so what we did is we took the same CD40 antibody and we treated a lymphoma model with the same CD40 antibody that had just been isotype switched to either be a mouse 2A or a mouse IgG1. And you can see here very clearly that the mouse IgG1 isotype in red basically delivered 100% cures in the mice, whereas the mouse IgG2A didn't deliver any therapeutic efficacy. So this was a real, I guess, surprise to people in the field at the time, and that's simply because when you compare that with a direct targeting antibody, such as an anti-CD20, like a rituximab type antibody, the profile is entirely the opposite. Those antibodies work by engaging activating Fc receptors with the mouse 2A isotype and don't work very effectively with the mouse 1 isotype. And so we had a very different paradigm here for immunostimulatory antibodies. And what we wanted to do really is understand the basis for that process. And so we, in the lab, and this was led by Anne White in the lab, and what she showed was that the mouse 1 isotype was able indeed to expand cytotoxic CD8 lymphocytes whereas the mouse 2A isotype was not able to do that. She then confirmed that that isotype was engaging the inhibitory Fc receptor for its activity by showing that in the mouse lacking that receptor in the knockout, there wasn't any expansion of the cytotoxic CD8s. So this showed us very clearly that there was a link between engaging with the inhibitory Fc receptor and expanding cytotoxic antigen-specific T cells. And that correlated directly with therapy. If we repeated the therapy that I'd already shown you, giving us 100% cures, and did that in the context of blocking the inhibitory receptor, then we lose all therapeutic efficacy. So this was a really important initial paradigm. And then the question then was, how is it working? And this experiment that Anne did here was essentially looking at the capacity of these different antibodies to drive B cell proliferation. And we use this a lot as a surrogate in the lab because it's a very convenient in vitro assay we can do. We can measure B cell proliferation. And what she was able to show was that the two different isotypes I've mentioned already, the mouse IgG2A antibody didn't drive B cell proliferation, whereas a mouse IgG1 did do that. So I should mention that the reason why we do these assays with B cells is because B cells express CD40 constitutively, so they're very convenient to look for B cell proliferation. But the surprise really was that when we then took a mouse IgG2A antibody and chemically cross-linked it, we were able to deliver the mouse 2A isotype with very high agonistic activity in driving B cell proliferation. So this was the first indication that it was actually the cross-linking of that antibody that was driving the heightened proliferation. And importantly, I think for the kind of general understanding of how these antibodies and receptors work, we were able to show in a variety of different models here using a vaccination model using ovalbumin as the target antigen, and then measuring antigen-specific T cell expansion, that if we did the same trick of class switching with either antibodies to 4-1BB, CD27, or OX40, which are other TNF receptor co-stimulatory molecules, we saw exactly the same profile, i.e. in every case, it's the mouse 1 isotype that drives the really nice expansion of these antigen-specific CD8 T cells. And then thinking mechanistically about how this was happening, we'd shown that it could be driven by further cross-linking. So then we started to think about receptor clustering, and we looked at that in two different ways. We generated some jerkat cells expressing human CD40 and then tagged with GFP. And what we were able to show is that when the cells are untreated, and then we just look at the distribution of the CD40 on the surface, it's distributed around the cell. When we add the native ligand, human CD40 ligand, you see these nice punctate clusters. When you give an agonistic CD40 antibody, you see this same clustering. And then we did the same experiment using antibodies to measure the clustering. So these were just Alexa 488 labeled antibodies. And what you see using either a non-agonistic antibody or an agonistic antibody, the agonists drive receptor clustering. And that's been a very clear observation throughout all of our studies on multiple different TNF receptors. So antibody clustering is what drives activity in each case. And thinking about this from a mechanistic point of view, all these TNF receptor family members have trimeric ligands. And so that means that individual monomers are brought together by the trimeric ligand, and then we get downstream signaling, which causes activation of our cell types. But thinking about what happens with an antibody, which is a bivalent molecule, it can't bring 3 receptors together, it can only bring 2 receptors together. So the paradigm was really that we thought the inhibitory Fc receptor was basically providing a clustering function able to bring those receptors closer together to drive the clustering that would give appropriate downstream signaling. And so that was one of the key properties that we know is required for an agonistic antibody to some of these co-stimulatory molecules. And we started thinking about what other properties are important. And one of the first things we considered was what's the role of epitope? Does it matter where on that molecule that we engage and bind? And because we'd been working on CD40 for some time, we had a whole antibody panel. Some of those were antibodies that we developed ourselves in Southampton that were agonists and had been into the clinic. Some of them were other antibodies, such as the CP87893 antibody that was developed by Pfizer. That was a very powerful agonist. But importantly, we also had a whole series of antibodies that were antagonists. So they were designed to block CD40 signaling. And again, they'd been into the clinic. So CD40 is a 4-domain extracellular protein. So it's got cysteine-rich domains on the extracellular surface, which form into these immunoglobulin-like folds. And what we wanted to do essentially was to then effectively assess where each of these antibodies was binding on the extracellular part of CD40. And we did that through a different number of approaches. We were lucky enough to be able to solve the crystal structure of our antibody Kylop-74 binding, and we saw that it bound in the very membrane distal domain, cysteine-rich domain 1. And that was in collaboration with crystallographer Ivo Chews and Chris Orr, and project that was led alongside with Anne White. But importantly, sorry, just to go back to that, what we were able to show was that our antibodies, essentially, that we've got from our panel, bound and decorated various domains across the whole of CD40, shown here with the ribbon diagram. So we then needed to, having demonstrated where these different molecules bound, we then wanted to correlate that with agonistic activity. And so that's what we did. And again, using the B-cell proliferation assay shown here, what you can see is as we got more membrane distal, we got greater activity. Importantly, in the B-cell assay, we're providing the inhibitory FC receptor that I mentioned was important for agonism, because B cells only express one FC gamma receptor, that's the inhibitory FC receptor, CD32. We were able to show that for the majority of antibodies, that was important for activity. So in the red bars, what you see is the activity when we knock out the inhibitory FC receptor. You see almost all those antibodies lose most of their agonistic activity, with the exception of this very interesting antibody CP, which retained high levels of agonism even in the absence of the ability of that inhibitory FC receptor to provide cross-linking. And then another important component of this study was the fact that we then introduced CHO cells expressing the inhibitory receptor in trans. And what we were able to show is that we were able then to drive incredibly high levels of B-cell proliferation if we provided CHO cells expressing, it has to be said, super physiological levels of that inhibitory receptor. And that, I think, says a couple of important things. And one of them is that epitope is important, that the inhibitory FC receptor is important for the majority of most of these antibodies, but that some of them, like the CP antibody, retain that activity in the absence of having that additional cross-linking function. But if you provide super physiological levels of this cross-linking, all of the antibodies become highly agonistic and you lose any subtlety of epitope. Importantly, we then went on to show that this B-cell proliferation activity also was mirrored by the ability to expand antigen-specific CD8 T cells using the ovalbumin system, as I mentioned already. Again, those antibodies that were membrane distal were the ones that drove agonism. And again, we had this CP antibody that was most active of all. So the summary here really is pretty straightforward, that we can have some epitopes that are more agonistic than others. So this CP870 antibody is what we call a super agonist. It's highly agonistic, but it also is able to do that in the absence of cross-linking by the inhibitory FC receptor. But more generally, that targeting the top of the domain, the most membrane distal part, was the agonistic region that we should aim for. The interesting thing about the CP antibody was that it was originally in the clinic as a human IgG2. And we wondered actually what would be the consequence of trying to develop this in terms of human isotypes instead of mouse isotypes. And unfortunately, although the two FC gamma receptor systems are very similar between mouse and humans, there are differences. And although we have activating and inhibitory FC receptors in both systems, when we look at the isotypes available to both species, none of them really mimic what the mouse IgG1 isotype does in terms of the human isotypes. So there isn't a human one, there isn't a mouse one equivalent in humans. So Anne basically took the pragmatic decision to essentially just assess all the human isotypes. And so she did that by taking our CD40 antibody, Kylop-74, producing it in the different isotypes you can see here, and then assessed for B-cell proliferation. So here you can see that the human IgG2 isotype was the most proliferative. It caused B-cell homotypic adhesion and drove B-cell proliferation the strongest. Interestingly, it did exactly the same thing in the ovalbumin immunization assay, so driving synfecal positive CD8 T-cell expansion the strongest. But But the real surprise was when we did the experiment in the inhibitory Fc receptor knockout mouse. And that was to show that we saw exactly the same level of agonism with human IgG2, and that was completely independent of the mouse inhibitory receptor. So this was a real difference towards what we'd seen before with the mouse isotypes. But we wanted to first see whether this human IgG2 property of being more agonistic for one of those CD40 antibodies could be used to leverage higher agonistic activity in general. And we thought the most challenging way of testing that was to see if we could take an antagonist and convert it to an agonist simply by isotype switching from human 1 to a human IgG2. So what we did, we took this antibody 341G2, which actually had been into the clinic as a human IgG4, and it was an antagonist because it blocks CD40 ligands. You can show here in the ribbon diagrams, this was some alanine scanning mutagenesis data we had showing where 341G2 bound. So it bound between CRD1 and 2. CD40 ligand binds basically in that same region. So you can see why when 341G2 had bound, it would block CD40 ligand from binding and so be an antagonist. And we formally demonstrated that using our B-cell proliferation assay, using CD40 ligand to drive B-cell proliferation, and showing that if we added either a human 1 or a human 4 antibody, we would block that B-cell proliferation and also block the humoral response in vivo, completely ablating the ability to generate anti-over antibody response. So we had a de facto antagonist. The question, what happens when we then convert it to a human IgG2? And what we saw was this incredibly large level of B-cell proliferation just by doing this isotype switching. So that was a real profound result. We went on to then examine this across a few other different isotypes and different antibodies. So you can see here a range of different CD40 antibodies that had been in the clinic, and comparing with human CD40 ligand as the positive control. Here's the 341G2 data showing completely inert as a human 1, strongly powerful, stronger than the CD40 ligand as a human 2. And you can see in a number of other cases that human 2 was driving more agonistic activity. And again, interestingly, the CP antibody seems to be independent of the isotype. It's still agonistic both as a human 1 and a human 2. So something special about that epitope. But importantly, we wanted to then explore whether this B-cell proliferation assay was also shown in the ability to drive CD8 T-cell expansion. That was exactly the case shown here. It was actually more profoundly agonistic than the CP antibody, which was the most agonistic antibody we've seen previously. It was able to do that independently of the Fc receptor, so knocking out all Fc receptors, we still saw very strong CD8 T-cell expansion. We made a number of antibody mutations, knocking out Fc receptor interactions. It made no difference to its activity. And then we did something to really explore what was special about the human IgG2, and I'll come back to that. And we essentially did a hinge swap between human 1 and human 2. And without going through the details, essentially what the conclusion from these assays was that as long as that antibody had a human 2 hinge, it was highly agonistic. And I'll come back to that later. The other thing that it was able to do was do what we anticipated in terms of mechanism of action. It was able to drive very strong dendritic cell activation, so measuring CD8 DEC205 dendritic cell activation in vivo. This is in an Fc gamma receptor wild-type mouse with the human 2 driving up activation markers CD80, CD86. If we then did this in an Fc gamma receptor null mouse, then we were still able to drive that. We showed that this was also similarly the case with human dendritic cells, monocyte-derived dendritic cells. Again, the human 2 shown in green was the most agonistic format, and equivalent to the ability to activate dendritic cells as something like LPS, with the human 1 isotype basically pretty much inert. But what we're trying to do really is immunotherapy. We want to be able to treat cancer with these interventions. And so what we started to do was develop a couple of models where we could study that. So this first model was in a thymoma, which expresses ovalbumin as a foreign antigen. So here we gave the tumor on day 0. We put in some antigen-specific OT1 T cells that recognize ovalbumin on day 7, then gave our CD40 antibodies on day 8. And then we bled on day 13, basically able to show that we were able to expand our antigen specific CD8 T cells in the presence of tumor, having given tumor a week prior. But more importantly, then we were able to very nicely control tumor with the majority of our mice basically surviving out past 100 days. And this was entirely isotype specific. We then did the second model, which is more of a peptide vaccination model. So this was in the TC1 model, which has the E6, E7 antigen driving the tumor. And so we have a peptide that is able to recognize and drive responses to those. And so in this model, we give tumor on day 0, then we give the peptide along with the stimulatory CD40 on day 5, 8, and 11. Then we bleed on day 12 and we look for antigen-specific T cells. We were able to show that they are indeed induced with the 34-1G human 2, and also in an Fc gamma receptor null mouse. So this is Fc receptor independent. And again, very nicely, we're able to control tumor in a way that's much more effective even than the human 2 isotype of the CP antibody I showed you before. But this trick of changing the isotype from human 1 to human 2 and driving agonism is only useful if we can do it for multiple antibodies. So we basically took another agonist, this antibody, and we converted it from human 1 to human 2. We were able to show that the human 1 didn't drive receptor clustering and that the human 2 did. It was able to drive B cell proliferation and then antigen-specific CD8 T cell expansion in vivo. And then obviously because 3 is the magic number, we did it for a third antagonist, this AbbVie antibody, again, as a human 1, not clustering, as a human 2, clustering very nicely, driving B cell proliferation and driving the expansion of antigen-specific T cells once again. So that was really nice to show that for 3 different antagonists, we could just do isotype switching to a human 2 and we get agonism. But how about other immune receptors? So we tried the trick for antibodies to 4-1BB, to OX40, both TNF receptor superfamily members, and showed the same thing. Able to drive clustering as a human 2, able to drive increased activation as a human 2. And then lastly, we also did this for a non-TNF receptor superfamily member. So this trick is actually not just something that is confined to TNF receptors. CD28 is an immunoglobulin superfamily member. And again, the same thing. We were able to drive nice clustering of the receptor and very profound increases in activity as a human 2. So that then really brought us back to the point about what's special about human 2. And we went back and kind of reviewed the literature about human 2. And one of the things that is known is that human 2 has additional disulfides in its hinge. So human 1 basically has these just 2 disulfides, and human 2 has 4 disulfides in the hinge. And Amgen had done some really nice work previously, and what they'd shown is that under redox conditions, actually you can have multiple forms of human IgG2, which they called the A and B as their kind of 2 polar opposites. And what you actually see is this disulfide shuffling. And that is, i.e., you're rearranging the disulfides between the different chains under various conditions. And interestingly, this is something that happens in blood naturally as you produce a human 2, typically as an A format, and it converts to a B format over time. So we thought this was a really interesting thing that the human IgG2 could do. And Anne had already shown in the lab, if you use chemical approaches to basically to skew your antibody to either an A or B format, that it was the B format that was able to drive agonism and not necessarily the A format. So just to summarize then, we know that human 2 is something that can drive immune receptor agonism. It's driven by receptor clustering. We know the hinge is central. I showed you those hinge swaps. And we know that the human 2 hinge is unique and can undergo disulfide shuffling. So what we decided to do to really probe that was to design a series of hinge variants with restricted disulfides. And by that, I mean we mutated the cysteines to serines so that they could or couldn't form certain disulfides. And so when we did that, we produced a series of antibodies with different mutations, and then we were able to show using B cell activation markers such as CD23 or hematopoietic adhesion or B cell proliferation, essentially that we got an activity series. You can see here, as we change these different mutations, we start to drive up agonism, that this one is clearly the most agonistic, and that correlated with its ability to drive B cell proliferation. So we've got a series of variants that span agonistic activities. The epitope is identical. All we're changing is the hinge so that there's nothing different about where these antibodies are binding. It's only the hinge that's different. So the question was, what are disulfides doing? And so what we needed to do is really position those disulfides. So Chris Orr was a PhD student in the lab whose job it was to crystallize these different formats, and he managed 5 of the 6 different variants. So this was the key component of his PhD. And essentially, the net sum of the crystallography was they all form very similar structures. We solved the structures as Fab2s, I should say, just because it's simpler to get the crystals to form. But the key thing that Chris did was then he did anomalous disulfide scattering. And that allows you to position the disulfides exactly where they're forming. So you can see where the sulfur bridges are between the different molecules. And what we were able to do with that was to show that actually is those non-agonistic forms basically formed what had earlier been predicted as the A-type format. But the B forms, but the forms that were agonistic rather, basically had this disulfide crossover here, which was correlated directly with the ability of these antibodies to be agonistic. So that was all very useful, but obviously X-ray crystallography is a static technology. It shows you a snapshot in a crystal lattice. What we wanted to do really is to understand a little bit more about how these antibodies might be interacting with receptors, which obviously is a dynamic process. And so for that, what we did is another approach, which is small angle X-ray scattering, which is an in-solution technique. It's a low resolution technique, but it allows you to see how these molecules tumble in space and their maximum dimensions and various other properties. And what that showed was essentially between our least agonistic format in red and our most agonistic format in blue, that there was a clear difference in the Sachs profile. So we assessed this using something called a Cragg plot. But there's without the time to go into that, essentially what we were able to show is that the agonistic variants which had the disulfide crossover basically had a smaller maximum dimension than those that were non-agonists. So essentially the agonists were more compact as they basically tumbled in space. So that gave us another insight into how these molecules might be operating. But what I think coming back to the question that Caetano asked at the start, how is this useful to be interdisciplinary? Essentially, we then engage with computational chemists who were able to do things like molecular dynamic simulations, which was taking the crystal structure, running models according to the laws of physics to see how these molecules might move, but then taking the Sachs data into account as well. So we're able to look at ensembles and look at how often they might adopt different conformations. And that showed us very clearly that the formats that had been shown in Sachs as being less compact here adopt multiple conformations and adopt multiple conformational space, whereas the more agonistic forms are much more compact and much more restricted in the conformations they adopt. And these movies you can see here basically show you the non-agonists and the agonists. And what you see is the agonist will flip and twist. It's much more mobile than the agonist at the bottom in blue. So the lack of flexibility is associated with fewer conformations and higher agonistic activity. So I think that was an important concept that's led to a number of different engineering projects in the lab. So the question then became, can the human IgG2 hinge restriction drive activity in other antibodies targeting other receptors? And so we did this for a 4-1BB antibody, showed using the same series of mutations that, again, we were able to show with the Cragg plot that these mutations drive more compact formats of the antibody. And indeed, that correlates with higher levels of activity with the most compact format, an intermediate format, and the least activity is seen with something that is most mobile. We've also very recently, in the last week or so, shown that the same is true for OX40 antibodies. So this concept of constricting the mobility of our antibody arms by hinge engineering basically gave us the question, like, can we actually take this even further? Can we restrict the hinge even further? And can we drive even even greater levels of agonism by doing that? And the idea was to use structure-guided design to engineer those antibodies. So this is something that Hayden Fisher did, who's was a postdoc in the lab. So he basically took the crystal structure we had of the most agonistic disulfide format, and then he looked at that and said, okay, where could we design additional disulfides to get even more rigidity? And so he made two different variants, one called the staple and one called the clamp. And then we made those molecules, and then we assessed whether or not those disulfides that we'd engineered did indeed form where we thought they would. Very nicely, we were able to solve the crystal structure, and using the anomalous scattering show indeed that the staple and the clamp formed exactly where we'd predicted to. And then went on to show that they were indeed very compact, and that they were very inflexible as we'd predicted. And that was based on various biophysical assessments, which we did at the time. We're also reviewing different ways of assessing that at the moment. But the key thing is whether or not we were able to get more agonistic antibodies. And essentially, by using the clamp or the staple, we got a more agonistic antibody, more effective at lower concentrations, so a lower IC50. But I think even more importantly, when we then went, so this is sorry, I should say this is a reporter assay where we're measuring NF-kappa B as the readout, which is seen as GFP positivity. When we did primary human B cell proliferation, we were able to basically take the level of agonism that we saw with our previously most agonistic human IgG2 isoform and drive even higher levels of B cell proliferation. So restricting the hinge even further was able to drive higher levels of agonistic activity. Just to summarize that part, hopefully what I've convinced you is restricting the human IgG2 conformational hinge basically via this disulfide crossover gives us higher activity for CD40, 4-MeB, and OX40. We can then use structure-guided approaches to engineer new antibodies with more that are more compact and more restricted. And then we can do that to get antibodies that are actually more agonistic. And we feel that this could be actually a very easily translatable method to improve the activity of a variety of different immunostimulatory antibodies to different targets. And so I'm going to switch now from the hinge. I've talked so far about epitope, I've talked about the hinge, but how about other properties of antibodies that might be able to augment agonism? And so what we considered, what were other antibody characteristics in the Fab domain, the binding domain, that might be able to modulate? And I've talked about epitope. So one of the key things about the Fab domains is affinity, how tightly it binds to its target. And we know for direct targeting in natural antibodies that we reduce in response to infection, that high affinity is key. That's why we have somatic hub mutation. That's what we have in trying to design our antibodies that will be very useful for fighting off infections and also for receptor blockade. So the checkpoint blockers, high affinity is key. But what about immunostimulatory antibodies? So to answer that question, what we did is we went back to our favorite CD40 antibody. We already had a crystal structure. So we used some techniques using PISA and PyMol really to design mutants that would allow us to drop affinity. So basically manipulating the interface here. And what we did is we designed those mutants, produced them, and then assessed them in Biocore for affinity. And you can see here that we had a whole series of antibodies going from high affinity down to very low affinity. And so we were able to then evaluate then in our activity assays, showing, looking here at CD23 activation on our B cells, you can see that there was a clear increase in activity as we drop affinity, peaking here with this particular mutant, and then dropping off again. So we have a bell-shaped curve of activity, which actually correlated very nicely with the idea about whether an antibody can stably bind at the cell surface. So obviously, if you mutate the affinity too far, you ultimately aren't really binding and interacting with the receptor. So we had this nice activity profile of dropping affinity and increasing B cell activation, measuring by CD23. That was exactly the same for CD86 and also B cell proliferation. So in each case, we had this nice bell-shaped curve of maximum activation at lower levels of affinity before the drop-off. But key to this was whether or not we could do the same trick in terms of leveraging CD8 T cell activation, and we were able to show the same thing, that as we dropped affinity, we got more active antibodies that were able to drive greater CD8 T cell expansion, and then again we have the drop-off in affinity. This was also true in the presence of tumor. We were able to drive better CD8 T cell responses with the lower affinity variant. And indeed, that was able to show better tumor control than our wild-type antibody in this tumor model. That correlated very nicely with the ability to form better clusters. You can see here this low affinity variant had much more clustering than the wild-type mutants. But the question then became, okay, we've done it for our favorite CD40 receptor and our CD40 antibody. Does it work for other TNF receptors? So what we did was we took a non-agonistic 4-1BB antibody that had been used in the clinic, utomilumab, and saw whether we could take the same approach. So again, using the structural biology guided approach, this wasn't our structure, this had already been published. We looked at the interface, we made some mutants, far fewer mutants in this case, but again, we had an affinity series which correlated very nicely with an increase in agonism as we dropped affinity. And that again correlated very nicely with the ability to form measurable clusters on the cell surface. So the question is, does the magic work for other receptors and other antibodies? The answer is yes. The question then became, does it work for other non-TNF receptors? So what we thought here would be, could we take a non-agonist checkpoint blocker, for example, nivolumab, and turn that into an agonist? So this was just proof of concept. Could we take another receptor, another antibody that was actually a non-agonist and convert that? So same approach, take the antibody again, not our crystal structure already published, make a nice affinity series, dropping the affinity, and then we were able to measure whether or not we got better PD-1 signaling. So remember, PD-1 is an inhibitory receptor. Using an NF-kappa B activation assay as a reporter, just simply we were able to show very nicely that we were able to drive increased PD-1 signaling with a lower affinity antibody. That also correlated with better clustering. You can see here the nature of the clustering is very different. We're getting very large clusters in this context, and that's something I'm very interested in terms of, you know, the cell biology of what's happening with these different receptors and different biology in terms of receptor clustering. But importantly, when we then put these mutants basically into assays that were then able to block T cell CD3 signaling, we had this very nice affinity series that the lower the affinity, the more we could block CD3 mediated signaling, and that correlated very nicely with the drop in affinity. So just to summarize that part of the work, hopefully I've shown you that the Fab affinity does impact receptor agonism for 3 different receptors. That increased agonism correlates with receptor clustering. And just to re-emphasize, that's true across all the work we've done over the last decade. Always for receptor agonism, you need clustering. We've been able to convert a weak agonist into a strong one, a non-agonist into an agonist, and an antagonist into an agonist just by modulating the affinity. And it offers, I think, a rational, efficient, and a highly tunable solution to deliver antibody-mediated receptor activity. And I think in the world we live in now, where there's an increasing ability to make predictions and solve structures in silico rather than having to do the crystallography. So Chris Orr, the PhD student, spent 3 years solving structures for us to be able to kind of move forward that part of the project. Now there probably are, or soon will be, methods that will allow us to kind of make those predictions and at least test mutations more rapidly than having to solve a structure and move on, which can be quite time consuming. So just to bring the whole thing back to the start, you know, the aim here is to drive agonism of different types of receptors. Predominantly, we focused on TNF receptors, but it isn't exclusive to those. If we think about what happens in nature, physiologically, typically what we're talking about is taking trimeric ligands that bind monomeric TNF receptors, and they bring them together, they cluster them, they drive binding of TRAFs or whatever adapter molecules are inside the cell that drive then subsequent signaling processes. So that's the natural situation. In the lab, I'm sure many of you have used secondary antibody cross-linking before, where you bind your antibody, then you come in with a secondary antibody to cluster that first antibody and thereby the receptors. And that's something that can be used. The cautionary tale there is, you know, whether that is physiological. The third example that I talked through at the start is the ability of Fc gamma receptors to mediate this cross-linking. And so they can do that. Predominantly, that's done in trans. That isn't work that I've had time to talk about. And so again, we feel that that's a clustering biology, that they're able to cluster the antibodies and the receptors. And the cautionary tale there is the experiment I mentioned, where if you add a CHO cell expressing super physiological levels of an Fc receptor, you can make any antibody appear agonistic. That doesn't mean that's what is predicting about what will happen in vivo in the real situation. Then I talked about the human IgG2 specific disulfide bonding pattern that we know can drive better levels of agonism. And we feel what that's doing in that case is actually it's restricting the receptors to be closer together. So even though it's only binding 2 receptors at any one time, it's keeping those receptors in close proximity. We feel that the bivalent nature means that one antibody arm may then come off and bind to a third receptor and bring it into the cluster. Because of the nature of the hinge, those clusters basically stay together to eventually nucleate nice functional receptor clusters. I talked about the epitope-driven mechanism. So in the case of CD40, this CP antibody basically works with any type of hinge, with any type of isotype. And so there's something unique about the epitope it binds, and we feel that somehow it must be able to cluster the receptors very effectively, and maybe somehow it mimics the orientation of the ligand. And that's something, again, I'm very interested in kind of trying to understand. Interestingly, with 4-1BB, there's a similar antibody urelumab, which has been in the clinic, which again seems to operate like a superagonist. It seems to work in multiple isotypes and multiple formats. And then lastly, I talked about the low affinity model, whereby we can reduce affinity and get better levels of agonism. And we think the model there is that it's like a sheepdog model, where you're trying to put a sheep into a pen, that you bind 2 receptors. Because of the low affinity, one arm comes off, it can then bind to a third receptor, it brings it into the cluster. The arm comes off again, it can bind to a 4th receptor and bring that into the cluster. And I should mention, I didn't have time to show the data, but essentially the key parameter in affinity that we've seen that correlates with activity is off rate. So it fits with that idea that it binds, releases, and that's the kind of driver behind that type of mechanism. So hopefully I've given you a kind of whistle-stop tour of ways in which antibodies can drive receptor agonism. Increasingly now, people are thinking about these things for inhibitory receptors. So I've talked about activating receptors, typically co-stimulatory receptors, but actually there are many immunological processes and pathologies may want to activate inhibitory receptors more strongly, not least of which PD-1. So I'll just kind of end in saying that the challenge really is discovering the optimal approach in humans in each case. So all of these technologies are available. Which ones should we use? And then targeting this activity to the right location. And that's a whole other sphere, is like, how do we make sure we drive agonism in the right place to get the right type of activity without any kind of toxicity. And then the most important slide last is to thank all the people involved. All this work was started in Martin Glennie's lab and led initially by Anne White. Then Chris was the postdoc who solved all the crystal structures. Ben came in as a fantastic postdoc and led the work that I mentioned around human 2, Epitope, and is now has his own lab in Westlake University over in China, taking some of these things forward. Hayden was a fantastic postdoc in the lab, taking forward a lot of the computational side of things and the modeling. And Izzy is the current PhD student, taking forward the Hinge engineering technologies alongside Cree. My partners in crime are John Essex, who's the computational chemist who does the molecular dynamic simulation. Ivo Hughes is the structural biologist. And in Southampton, we have a fantastic group of PIs that are all interested in antibody biology in general, but particularly around TNF receptors. So Eamon, Steve, and Sian. We have a fantastic team of people that make all of these antibodies. So they're a long-suffering team. I can come up with 20 antibodies overnight that they very happily will make, or begrudgingly will make. And then obviously there's multiple people around the world, multiple beam lines that have helped us collect the structural biology work. So I'll end there, and I'm very happy to take any questions.
Well, thank you very much, Mark, for that fascinating lecture. Just to remind everyone that you can ask Mark questions offline, and the instructions are up on your screen right now. They were also pasted into the chat. And so it just remains for me to thank Mark once again for taking the time to share his research with us. Thank all of you for participating in the Global ImmunoTalks series. do remember to come back week after week. Next week, we've got a lecture by June round. And if you know of anyone who might have missed any of the Global Immunotalks and wishes to catch up, just remind them that they are all available online as recordings. So thank you again, Mark, and thanks to all of you, and see you at our next Global Immunotalks edition.